Today we're going to be talking about protein synthesis. That is actually two processes, both transcription and translation. And we are going to follow the target that we want you to be able to describe and model how information are DNA codes for creating proteins. Alright, so there's a few questions you'll want to know. Where is the DNA located? When translating, which of the following is not true? True or false, DNA codes for how to make proteins. What is the shape of RNA? Proteins are synthesized. This is a three-letter word found in the mRNA. True or false, there's only one codon for each amino acid. Which molecule carries amino acids to the ribosome? mRNA can enter the nucleus. True or false? And you're going to want to be able to turn a sequence of mRNA into amino acids. So that you should be able to do all by the end. So the first process is transcription. And with transcription, we are taking the DNA, which is this double helix model right here, this DNA molecule, and we are turning it into mRNA right here. All right, so that's a process that we're going to learn about right now. The first thing we need to do, we need to unzip this DNA, just like you see right there. And we use, again, enzyme to help us do that. RNA polymerase is going to help us out by putting down these new nucleotides. The RNA polymerase pairs RNA nucleotides with one side of the DNA strand, like you see right here. And very similar to what we learned in replication, there's base pairing rules. DNA nucleotides, if here's our nucleotide, A is going to turn and complement U. Thymine is going to complement adenine. Guanine is going to complement cytosine. Cytosine is going to complement guanine. So U stands for uracil. So in RNA we have uracil, where in DNA we have thymine. So there's no thymine over here, and there's no uracil over here. Our DNA makes a long chain of mRNA, right? It's just one single-stranded molecule, mRNA, made of nucleotides again. And that is not permanently attached. It's going to be loose. So we start here. Here's our DNA. It opens up. We start making mRNA, and then it's going to detach. After that, zip backs up, and we have our molecule. So, not what is made, but what is made in transcription. What is one difference between RNA and DNA? Make sure you're able to answer those. So look back and find that out. And let's go ahead and watch this little video.
The cell transcribes the genetic designs for each protein from strands of DNA to special molecules of RNA. An enzyme unwinds a segment of DNA, making room for its complementary RNA nucleotides. Both DNA and RNA nucleotides contain a sugar, phosphate, and base. But in RNA, the sugar is ribose, and one of the four bases is replaced by uracil. The exposed bases of the DNA strand serve as a model for the construction of messenger, or mRNA. On other strands, the cell produces transfer, or tRNA, and ribosomal, or rRNA. In their final form, these molecules play a vital role in protein synthesis.
Alright, so that kind of showed transcription. Transcription, again, is the process of making mRNA. So, some things we need to know. What is a triplet or codon? So a triplet is a three-letter code. It's called a codon if it's in the mRNA, triplet if it's on the DNA. So our codon, UAG. We can use this three-letter code to find out what amino acid we're going to end up using. So this is our codon wheel right here. So if we have UAG, let me get a pen, pen, UAG, we're going to have UAG. So that's stop, right? CUU, CUU, that'd be leucine, right? So these three-letter words pretty much are going to give us an amino acid. So there's a few you should know. You should definitely know AUG, the start codon. Sometimes instead of the codon wheel, you'll see this little graph, but same thing. The first letter, A. The second letter, U. So we're in this box right here. AUG. Hey, what do you know? It's methionine, or the start codon. There are some codons that are nonsense, and if you'll notice, also, there are many amino acids that have more than one codon code for them. So it's not like a one-to-one. There's a total of 20 codons altogether. 20 codons altogether. Oh, not codons. 20 amino acids. Sorry about that. Alright, so what we need to do now is practice changing this DNA sequence to mRNA. So you have AAC, AAC. We're going to go to AAC. So that'd be asparagine. Oops, I made a mistake. Maybe you didn't make this mistake. This is a DNA code. Alright? I did this on purpose, of course. Our DNA code, we don't want DNA to use this wheel. We want RNA to use this wheel. So we need to turn AAC into UUG. And then GCC is going to go to CGG. I'll kind of stop there. So UUG. 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 Leucine. Alright, so our first would be leucine. Then our second would be CGG. CGG. Arginine. Alright? So our second would be arginine. Go ahead and continue practicing that. So, where is mRNA found? Where are proteins made? Both of these are really important ideas. What part of the cell are proteins made? Go look that up. I'm not going to tell you. What are proteins made of? Right. So now we're to our translation portion. Remember, first part was transcription. Second part's translation. This happens at the ribosome, right? Remember, the ribosome is made of ribosomal RNA, and there's two different parts or three different parts of the ribosome. I'm not going to ask you to know those different parts or the different sites. You just need to know that this is all happening at the ribosome. The mRNA is the instructions. So we have our DNA, which has the code. The mRNA takes that code from the nucleus out to the ribosome, and then this mRNA is going to code for our amino acids. So mRNA leaves the nucleus. Here's our nucleus. It's going to go to the ribosome. That's that blue and brown blob here. And these tRNAs, so this is a tRNA, bring amino acids to our ribosome, and they hook these up in order. Notice here I have a codon, these three right here. So the opposite of a codon is an anticodon, and notice how the shape fits. This one hooked up its amino acid, and now it's leaving. And again, we made a bond, so we lost water. Make a big polypeptide. So a polypeptide is a series of amino acids. We get enough together and we'll make a protein, but just having 5 amino acids in a row doesn't necessarily make a protein. And eventually it's going to stop. This is the stop codon. The stop codon doesn't code for any amino acid. All right, the ribosomes are the tool that helps attach them. Here's a little picture showing the whole process, starting at DNA, making our mRNA. mRNA goes out of the nucleus. We have our ribosome here, attaching those together, and we get our series of amino acids, which is going to make a protein. Here's another picture. The ribosomes attach to the mRNA at the start codon and falls off at one end of the 3 stop codons. Said a lot. Sorry about that. So, what is this molecule? What does it kind of look like? What letter kind of help you out? Here's another hint. Anticodon. What molecules have anticodons? What's the name of this process? I'm taking mRNA and making amino acids here. So, what process is that? And we will finish up that video.
Transfer, or tRNA. Translation begins with the binding of a messenger RNA to a small subunit of a ribosome, the cell's workbench for protein assembly. When properly positioned, the messenger RNA triggers the approach of a tRNA, which carries the first amino acid. The tRNA attaches only if its 3 nucleotides exactly match the first 3 coding nucleotides of the mRNA strand. A large ribosomal subunit now joins the group to form a functional ribosome, with 2 binding sites built from ribosomal proteins and rRNA. Soon, another tRNA arrives, which matches the next 3 nucleotides on the strand. With the help of the ribosome and some cellular energy, the neighboring amino acids bond together. The first amino acid then separates from its RNA taxi, leaving the ribosome in search of another, identical amino acid. Now the ribosome moves along the mRNA strand. This exposes the next set of nucleotides, which match those of another tRNA. With each amino acid, the protein continues to elongate. As it grows, it folds into the 3-dimensional shape so crucial to its function. When the process is complete, the ribosomal fragments separate, free to join again later. With a legion of these molecules in operation, a single cell can produce hundreds of proteins every second.
Alright, so now this should be easier. Where's DNA located? Is it located in the nucleus, the ribosome, the cytoplasm, or the Golgi apparatus? It is in the nucleus. When translating, which of the following is not true? So translating is turning mRNA into proteins, into a series of amino acids. So we're outside the nucleus, so we're not dealing with DNA. So this is not true. We're not turning thymines into adenines because there's no thymines that we're dealing with. Also, E should also not be true because we're not dealing with thymines. True or false? DNA codes for how to make a protein. This is a huge idea. You better know that it is true. What is the shape of RNA? It's not a double helix. It's not a pentagon or hexagon, single-stranded. Proteins are synthesized in the nucleus, at the ribosome, outside the cell by DNA. DNA does have the code, but that's not what makes it. They are ribosomed, or they are made at the ribosome. The 3-letter word found on mRNA, and that's real important, that's mRNA, is going to be codon. It would be anticodon if we said on tRNA. So codon is our right answer here. There is only one codon for each amino acid. That is false. Which molecule carries amino acids to the ribosome? tRNA, that's super important. mRNA can enter the nucleus? Absolutely. I guess more correct it would say it's leaving the nucleus, but you will find mRNA in the nucleus because that's where it's made during transcription. And which amino acid sequence is coded for by the following DNA sequence? Remember, this is our DNA sequence, and I'm going to let you figure that one out. Thank you. Hope you did a good job. Make sure you write your questions and your summary on your Cornell notes. We'll put these in your notebook, and we'll have some fun in the lab tomorrow.